All right, I think we're ready to go. It's pretty much smack bang on 1 o'clock. Thank you for joining us today. Before we start, I'd like to acknowledge the traditional custodians of the land on which we're meeting today. That's the land of the Wurundjeri people of the Kulin Nation, and I'd like to pay my respects to their elders and families past, present, and also emerging. So my name is Clover Maitland. I'm the Research and Evaluation Manager at the Cancer Council in Victoria for the SunSmart program. I sit in our Centre for Behavioural Research in Cancer. I'm relatively new to the role and I'm relatively new to Melbourne, so it's been fantastic to come along or be invited today to MC this event, as I've learnt quite a lot about the centre, and also I'm learning a lot about skin cancer and melanoma research. My background is in health promotion research and evaluation more generally, so I'm relatively new to skin cancer professionally, but I do have a really personal experience having lost someone to melanoma, so it's definitely a topic that's very, very dear to my heart, and I'm personally very appreciative and interested in the fantastic work that the research team do here, and very much looking forward to what they have to add today. So the Victorian Comprehensive Cancer Centre is a powerful alliance of 10 successful Victorian organisations that are committed to cancer control, and its role is to coordinate that collaboration between its member organisations and also to develop a strategic program of work that integrates research, education and also clinical care to maximise patient outcomes in a cost-effective manner. So they're doing a lot of things and doing it very well. In today's presentation, we've got some really interesting speakers coming up, but also I'd just like to draw your attention before we start to the program that is also coming up over the next few weeks, which you can see up there. So please register your interest at the VCCC website to attend those coming up as well. Part of the reason that I'm here today to MC is that we're acknowledging National Skin Cancer Action Week. So National Skin Cancer Action Week really provides a focus for the Cancer Council Australia and the Australian College of Dermatologists to promote the sun protections and sunspot messages, and also focus on early detection for people detecting melanoma early. David has just been speaking to me in the lead up, and he said that he really likes the new sort of slogan of the week. And this week it's about owning your tone. So not being, I guess, not being worried about tanning, especially for our young people, which we know don't participate in sun protection behaviours as much as our older populations. So it's really trying to bring the message home to them this week as well. So I guess to get the presentations that you're all here waiting for started, I'd like to welcome David Gyorki, Dr. David Gyorki, and also Dr. Stephen Wong from the Peter MacCallum Centre Cancer Centre. Now I've got a little bit of a spruiking here for you, David, before you get up. But David is the VCC Research and Education Lead for melanoma and skin cancers. In his position, he brings together multiple disciplines, organisations and perspectives to provide outcomes in melanoma and high priority non-melanoma skin cancer. He completed his education and training at the University of Melbourne and St Vincent's Hospital, and during this time he also gained an MD degree at the Walter and Eliza Hall Institute. In addition, he's completed a clinical fellowship in complex surgical oncology at Memorial Sloan Kettering Cancer Centre, and previously held the Herman Fellowship in Clinical Cancer Research at the University of Melbourne, and currently holds a Peter MacCallum Clinical Researcher Fellowship. Stephen Wong, who will speak to us second today, is a senior medical scientist and postdoctoral researcher who specialises in cancer genomics and molecular diagnostics at the Peter MacCallum Cancer Centre. And his research focuses on identifying and characterising novel molecular biomarkers in melanoma and other malignancies. So today, David will start off by giving us an overview of melanoma research here at the centre, and then later tell us a little bit more about his research before Stephen will present on making an impact on melanoma patient management through liquid biopsies. So could you please join me in welcoming David to the microphone.
Okay, thanks so much for that very kind introduction. Okay, so I thought that I would start off by just talking about melanoma research across the VCCC. just to provide a little bit of an overview before Stephen and I get down to the nitty-gritty of our own particular research interests. So it's National Skin Cancer Action Week, so this needs a little bit more time, because I think it's important that we all recognise how prevalent and how important a health problem skin cancer is. So 2/3 of all Australians will be diagnosed with skin cancer by the age of 70. Melanoma represents a relatively small proportion of that, but deaths due to skin cancer are overwhelmingly represented by melanoma, which makes up about 4/5 of the deaths related to skin cancer in Australia, which is about 2,000 or a little over 2,000 patients a year. So it's a big problem. And I think the most important thing to note about most skin cancers is that they are preventable. And so while both Stephen and I focused in our research on improving treatments for patients with melanoma, it would be much better if patients didn't develop melanoma at all. Now, not all of them are preventable, so I don't want it to sound like I'm blaming patients for their own actions in developing melanoma, but there is there has been historically a complacency in Australia about the risks of sun exposure, and I think the Cancer Council is doing an excellent job of bringing attention to that. And like I said earlier, I think the current slogan and campaign of owning your tone to avoid a tan is certainly a great message, which you are now all welcome to share across the community. So melanoma research in the VCCC is extensive. Melanoma has been a very productive group here at Peter Mac, but also at the Olivia Newton-John Cancer Centre and across the road of the Royal Melbourne. And I'll just provide a little bit of an overview of of what sort of work has been done in this field. So in the clinical setting, there's been a strong focus, particularly here at Peter Mac, in early stage melanoma, with trials in surgery for patients with early stage disease. And actually, we are opening a trial called MELMART this Friday, which is an international trial which hopes to set the standard of care for the extent of margins required for the excision of primary melanoma. It's a relatively pedestrian sounding topic, where we're comparing taking 1 centimetre of normal skin around a previous melanoma to 2 centimetres of normal skin, which is the standard of care. But that 1 centimetre can sometimes increase the complexity of surgery, and we think is probably an unnecessary centimetre and would save the community large expense, patients significant morbidity, and hopefully be an impactful study if it turns out that our hypothesis is correct. There's been a lot of work in locally advanced melanoma. Peter Mac led actually only randomized controlled trial of patients receiving radiotherapy after surgery for melanoma, demonstrating that the radiotherapy had very little benefit, and it's been completely practice changing. We are now looking into the neoadjuvant space with drug treatments that's before surgery to try and reduce giving drug treatment before surgery to try and reduce the risk of the cancer recurring. Stephen, maybe we'll talk about some of the work that he's done in some of these trials, looking at novel ways of monitoring patients. And then in advanced melanoma, many of the landmark studies which have defined the way that melanoma is now treated internationally have had significant leadership and participation from medical oncologists here at Peter Mac, as well as at the Austin Hospital and Olivia Newton-John Cancer Centre. There's a strong focus on patient experience research, and particularly around the patient experience of receiving immunotherapy, given that melanoma was the poster child for the wave of immunotherapy that's now being used in many cancer types. We've really been involved in some important work to identify what patients' needs are with these novel therapies, and then survivorship research has also been played a part. It's a strong translational research program looking at novel biomarkers, including circulating tumour DNA, which Stephen will talk about. There's work going on in autoantibodies at the Olivia Newton-John Cancer Centre and other immunological biomarkers. I'll talk a little bit later about understanding the immune microenvironment in melanoma, and there's a strong computational biology program also. And then basic research and fundamental discovery is critically important. We've got a major focus on that here at Peter Mac as well. So one of the drivers behind this successful work and collaboration has been a project called Melanoma Research Victoria, which was initially the Melbourne Melanoma Project, but was renamed about 3 years ago now. And I think Sonia Mailer, who's the executive of Melanoma Research Victoria, is in the audience. So thank you, Sonia, for your work. So Melanoma Research Victoria is a collaboration between the three largest volume, started off as a collaboration between the three high volume melanoma centres in Victoria, which were Peter Mac, the Alfred and the Austin Hospital, who together decided to create a combined database and biobank, which now has over 3,000 participants and has contributed, as you can see here, over the last 9 years to the development of an enormous and hugely productive resource that's used by a number of researchers across these institutions. It's multidisciplinary with inputs from a variety of different clinicians and researchers. And as I said earlier, there have been now over 3,000 patients recruited, mainly from here at Peter Mac in blue and the Alfred Hospital. And we have complementary clinical services. So the Alfred has a strong focus in early melanoma, and Peter Mac's focus is a little more in advanced melanoma. So those recruitments are actually very complementary. There have been a number of recent projects approved in medical oncology, surgery, radiation, translational research, including international collaborations. The IMDDP stands for the International Melanoma Discovery Data Platform, something like that. It's a big international collaboration that's trying to help define some key questions in melanoma, and there are now strong collaborations going on with industry. I'll just use this one collaborative example. So the AJCC, the American Joint Commission on Cancer, defines the staging system that's used for all different cancers. And that's the way that we box patients into various prognostic groups. And the melanoma, the most recent, the 8th edition of staging for melanoma, is actually the first of these staging programs that is completely driven by high quality data. So what this group did, which included about 2,000 patients from Melanoma Research Victoria, was to compile a data set of 40,000 patients and to actually look at the outcomes of those patients and to find out which particular characteristics best separated out the patient's outcomes. And so we now have a very high quality prospective data set that has helped us define how to stratify our patients' risk of recurrence and therefore how to optimally select their treatments. There's a strong group of consumers who provide input into Melanoma Research Victoria, some of whom are here in the audience today. We have a effective governance committee with representatives from all of the sites as well as from the consumer reference group. These are the partners and collaborators. And I'll just mention, this is aside from the MRV, the Melanoma Grand Challenge, which has been a project that that I've been working on with strong input across the VCCC. And when Grant MacArthur was the research education lead in skin and melanoma, he invited big questions in melanoma to be submitted a grand challenge. And Andreas Behren, who's a immunologist at the Olivia Newton-John Cancer Centre, and I put together a question in biomarker research, trying to use immunological markers to better stratify the risk of recurrence for patients having curative intent surgery. That group subsequently expanded to include Tori Ma from the Alfred and Tony Pappenfuss from here at Peter Mac and Weehai. And we've been working on this question. We put in an unfortunately unsuccessful NHMRC grant and have now been trying to redefine the question. And I just thought I'd talk you through the process that we've used, because I think it's been a really interesting and rewarding way to try and understand what is the key clinical question at hand and how to refine a study question. So we decided that the key area that we wanted to focus on was in melanoma biomarkers. And so we ran an largely educational symposium inviting various researchers from around the country to discuss their own specific areas of biomarker research. It was a one-day symposium. It was well attended, and I'm happy to report that we received very positive feedback from the audience about what they got out of it. But then we finished the symposium with a working dinner, which was attended by some of the key participants, but also by strong consumer participation, to try to identify what the key research questions were that remained. And my big thing that I took out of the day, but also what I had thought about leading into it, is that we often see research presented where a particular marker is identified that separates out patients who do well from patients who do less well, but actually very few of those markers actually make it into the clinic. So you can open any medical journal at any time and read a paper that says this marker will identify patients who are twice as likely to die from disease than patients who don't have that marker. But of those markers that are identified, it's an extremely small percentage that actually ever get to the clinic and are ever applied to patients in the real world. And so the question is, why is that? What is it about a marker that will or will not make it into the clinic? And the reality is that it's only going to make it into the clinic if it applies to a clinically relevant question. Purely identifying risk of relapse or risk of dying from a particular cancer is not impactful enough to take a biomarker into the clinic. And so where we're at now is we're working through what exactly the question is that we want to apply our biomarkers to. So that's been a very helpful process. And I will stop there and hand over to Stephen, who will talk about some of his research, and then I'll come back and talk about some of my research later. So maybe if we save questions for the end. You happy with that? Yeah. Perfect.
Thank you, David. Okay. So today I wanted to talk about some of the liquid biopsy research that we've conducted over the last few years, particularly in melanoma. But I thought I wanted to start off by looking at, first of all, about tumor biopsies. So this is where, a patient comes in, gets a needle inserted at the site of the tumor, and that tissue is extracted and histologically examined. Now, this is important because it's important to diagnose what type of cancer an individual may have, particularly providing staging information about the prognosis as well as treatment plan for the patient. Currently in this day and age with advances in genetic sequencing, we can also take that tissue sample and sequence it and find mutations which may direct targeted therapeutics. In the context of melanoma, we know that 40% of patients have a mutation in the gene called BRAF. And identifying that mutation is key in selecting either a targeted therapy regimen versus an immunotherapy regimen in the metastatic setting. But of course, there are obvious disadvantages to tumor biopsies. Foremost, they're very invasive and can cause significant patient discomfort. In the context of genetic sequencing, this can often take a long turnaround time, often requiring a tumor block containing the tumor to be retrieved from a pathology lab And this can sometimes take weeks, if not months. But importantly, tumor biopsies cannot be frequently collected throughout treatment. And this is important to note in melanoma because melanoma, just like many other cancers, are not static entities and they can evolve over time, requiring new mutations as they evolve. Consequently, if we were just to biopsy, as you can see in this slide here, one area of the tumor, we may miss important genetic alterations which may have impact on the patient. This is why I'm really excited about the recent advances in liquid biopsies, okay? Because this is where we can provide a non-invasive biomarker which can be collected frequently and can allow interrogation of a cancer patient's genome over multiple time points throughout their treatment. So what are liquid biopsies? Well, they come in many different shapes and sizes. I've just shown this slide here, but basically you can have things such as circulating tumor cells, circulating microRNA, and exosomes, all these microvesicles that are released from the cancer cells. And they can be collected from a variety of different bodily fluids, including urine, saliva, cerebral spinal fluid, but most commonly from a blood test, simple blood test that we already know forms part of standard patient management. My work over the last few years has focused on one particular type of liquid biopsy called circulating tumor DNA, or a ctDNA for short. ctDNA is very interesting because it carries the same mutations as those derived from the cancer cell. Consequently, if we are able to interrogate these mutations, we can potentially use it as a personalized biomarker for patient care. So what are some of these characteristics about ctDNA? Well, they're very short in size. They're about 170 base pairs in size. So this is often reflected because of the fact of how they're released into the circulation, thought to be through a process of apoptosis and cellular turnover as cancer cells grow and divide. They have a very short half-life once they're released into the circulation of about 1 to 2 hours. But importantly, the ctDNA is not analyzed in isolation, but rather is actually in a mix of normal cell-free DNA coming from other healthy tissues that are also turning over in the body. And actually, ctDNA only represents a very small abundance of the total cell-free DNA in a plasma sample. So what does that mean? Well, it means that we have to have very sophisticated and very sensitive technologies to find that ctDNA in a blood sample. It's kind of like finding a needle in a haystack. And so that obviously have implications about what type of techniques that we kind of use to analyze ctDNA. These include very standard laboratory techniques that we commonly use in a diagnostic setting, such as PCR. In the lab, we use something called digital PCR, which is very sensitive, but also can range into next generation sequencing approaches, such as targeted sequencing and even whole genome sequencing, which can interrogate the entire landscape of a cancer patient's genome. And while there's a trade-off between the sensitivity both in depth and breadth, there's two critical bits of information that we can collect from the analysis of ctDNA. One is a quantitative measure. You can measure the levels of ctDNA. And we know that patients which have high disease burden usually have high levels of ctDNA. And so with that knowledge, we can actually use ctDNA measurements as a way of monitoring disease over time and treatment. The other bit of information that we can collect is a genomic characterization of the cancer genome. And because we can collect multiple samples over time, we can map the evolving nature of the cancer genome over time. And this will provide very important insights into things like resistance mutations that could be acquired through treatment. So how do we process a ctDNA sample for analysis? Well, I just put this slide up here as a kind of like a rough workflow. So basically, a patient comes into clinic, we usually get a blood sample, usually about 10 to 50 mils of blood is collected. The blood is then usually spun down in the lab, and then the plasma fraction is only stored away in the freezer. When ctDNA is initiated, testing is initiated, we then usually extract the plasma DNA and then plug it into one of these sophisticated sequencing machines here. Once the results are analyzed and interpreted, they're fed back to the clinician and patient, and then that can direct patient management. Now, it's important to note that this entire process takes a matter of days now, as opposed to tumor-based sequencing or approaches where that can take upwards of sometimes of weeks. So what are some of the clinical applications of ctDNA? Well, I think this is something that it's really exciting because I think in the terms of melanoma, we can apply ctDNA to multiple facets of clinical management. When I started this work out a few years ago, I initially focused on metastatic melanoma. And I wanted to assess its clinical validity as well as utility in this setting. And we found a great number of findings, but I wanted to highlight just 2 cases where I think will demonstrate some of the impact that these kind of tests can have on patient management. So in this first case, I show a patient with metastatic melanoma. As you can see on the PET scan on the left, this patient had a pretty dire prognosis with multiple sites of metastatic disease across his body. The standard of care here is to take a tumour biopsy and to test it for BRAF mutation, because as I said before, if you have BRAF mutate, you probably get targeted therapy as your treatment option, versus if you're wild type, you'll probably get immunotherapy. Unfortunately, after multiple rounds of molecular testing, the test completely failed, probably because the sample that we collected was very degraded. So we therefore turned to using ctDNA testing as an alternative. And in the middle, you can see the many droplets were found positive in the blood sample for this patient containing a BRAF V600K mutation. So with this knowledge, the clinician was very confident in prescribing a targeted therapy for this patient. And as you can see on the PET image on the right, the patient responded very nicely to therapy. I think this case really nicely illustrates that therefore that ctDNA can be used as an alternative to tumour samples, particularly when it's unavailable or difficult to access. What's really amazing about this case is that we were able to turn this test around in time within 24 hours. So what about disease monitoring? Well, I'll highlight this case here where we've been able to take ctDNA testing in a serial manner and chart the levels over treatment. And as you can see over the corresponding radiological PET scans on the top, you can see that the levels nicely mirror those seen in PET scans. This means that you can potentially track disease and use PET scans, use ctDNA scans as an alternative to imaging in some cases. Now, while I don't think we'll ever replace PET scans because they're very, very sensitive, I think ctDNA will have a nice and powerful complementary role to these techniques in the future in terms of guiding when to image, as well as in the interpretation of results when they're difficult to interpret, such as when you've got pseudo-progression, where it's hard to differentiate between inflammation versus true disease. And I think this will be very important for patients who live in, for example, in rural areas where access to some of these imaging facilities is often difficult. And we can actually apply the ctDNA in order to guide when they should come in to get their scans. So what about early stage melanoma? What about stage 3 melanoma? Well, I think this is where ctDNA could be applied and make a great benefit in terms of the long-term morbidity as well as mortality of melanoma as a whole. So stage 3 melanoma is just disease confined to the primary side of the disease and the lymph nodes. And for a very long time, the mainstay of treatment was surgery. But unfortunately, patients after surgery have a very highly heterogeneous disease course, with relapse rates ranging from 30% to 90% depending on the substage. And despite curative intent and many patients experiencing clinical remission, we currently do not know which patients are cured through their surgery alone versus those that have minimal residual disease, that is a persistence of small number of tumor cells left behind that remain undetectable and then have a higher chance of relapse. I use the analogy here of a sheet of glass, for example, accidentally dropped on the floor. And while it's very easy to pick up, those large pieces of glass on the floor, it's those tiny little fragments of glass that are left behind, which may be very hard to see by the eye, that may be undetectable. And that's the same kind of challenge that we face in stage 3 melanoma. Currently, the way of detecting minimal residual disease is through surveillance imaging, but optimal follow-up strategies have not been clearly defined. And while PET is very sensitive at detecting minimal residual disease, it unfortunately can't be done on an ongoing basis because of cumulative radiation exposure, the increasing chance of false positives, and increasing costs of the healthcare system. Importantly, patients can now be prescribed adjuvant therapy in the stage 3 setting, including targeted as well as immunotherapy. But currently, we don't risk stratify which patients should receive it or not, based on their risk of relapse. For example, if you have a high chance of relapse, you're probably more likely should be receiving adjuvant therapy to mop up any minimal residual disease. But if you have a low chance, you're cured through surgery alone. You don't need it, and you can avoid the potential expensive costs as well as toxic side effects of treatment. So this is where we turn to looking at ctDNA as a minimally invasive biomarker to identify those patients which have the highest risk of relapse and potentially could be guided for adjuvant therapy. So I just want to say that this work was conducted by one of my PhD students, Lavinia Tan, who did an outstanding job in this and this was just recently published in Annals of Oncology this year. So what we did was we took a patient cohort from the Melanoma Research Victoria cohort, and we took tumor samples and then sequenced them for mutations. And then we designed personalized droplet digital or ctDNA assays for each of our patients. And then we analyzed if we could see detection of ctDNA in the blood sample prior to surgery, as well as after surgery and during the surveillance period, during which case they were closely monitored through PET scans. Any results were also validated through an external collaboration with the Christie NIH Foundation in Manchester, UK. So what do we find? Well, it just shows these two survival curves here. Now, these are based on the distant metastatic-free survival of our patients in the cohort. And basically, on both cases, both using a sample for prior to surgery as well as after surgery, detection of ctDNA was associated with inferior distant metastatic-free survival. That means they had the highest chance of relapse. In fact, when we looked at just the post-operative ctDNA sample, as shown in the yellow line here, and on the figure on the right, you can see that every patient that we detected ctDNA using this blood sample relapsed within 18 months. So this potentially allows the powerful role of ctDNA to potentially predict relapse and give the adjuvant treatment to only those patients shown in the yellow line, avoiding those patients which probably have a lower chance of relapse shown in the blue. So what about ctDNA as a surveillance tool? Well, I show this diagram here where you've got a collection of patients which have all had serial ctDNA performed up to the time of first clinical relapse. And as you can see, those patients that have a ctDNA detected, shown in a solid blue line, we were able to detect on an average a median lead time of 2 months prior to clinical relapse in many of those cases here. This happened in about 50% of patients. This suggests that in the future, if we use ctDNA as a surveillance tool, we could actually use this as a way of bringing up imaging closer in terms of their time point and allow detection of disease and therefore early intervention for treatment. The next thing that we wanted also to assess, and I'll just show this diagram here of a patient on a immunotherapy or immune checkpoint inhibitor trial, is to discover if ctDNA could have an impact, look at an impact of immunotherapy on adjuvant immunotherapy on these patients. In this particular case study, I show a patient where the ctDNA, as shown in the red line, was clearly evident after regional node clearance of the disease. This patient therefore potentially had minimal residual disease. After starting adjuvant treatment, the ctDNA levels rapidly disappear. So consequently, this kind of case study represents the fact that maybe in the future, ctDNA could be a very powerful biomarker to guide which patients would receive adjuvant therapy, as well as look at the impact of that adjuvant therapy on levels of disease over time. So what's next in this technology? Well, I think there's very exciting things coming in the next few years in this field. More knows there's going to be new technologies which are going to be more sensitive and have increasing use of multiple markers such as exosomes and proteomic-based approaches. And this combined approach will increase the likelihood of detecting melanoma in its early stages and potentially overcoming disease progression. But I think the important thing about this is that more and more we need more clinical trials that incorporate ctDNA into their design. And I've been very humbled, and it's really exciting that there's a new study starting on next year called Detection, where it will examine in stage 2 melanoma whether or not the treatment using adjuvant immunotherapy will be beneficial for long-term survival based on this, and they'll be guided based on the ctDNA results. in the future, I hope more and more trials like these will open up, and I hope really ctDNA will become a common practice for all melanoma patients worldwide. So with that, I'd like to just thank the group that I'm involved in, particularly my PI, Sarah Jane Dawson, but also I just want to thank all the patients and families involved in these studies. These important studies are not possible without your generous support, including the patient sample materials and stuff. Thank you very much. And after that, I hand over to David.
Great, thanks, Stephen. That was an excellent talk. And I'm going to change gears now and talk about some of the work that we're doing, understanding the immune environment in melanoma. And I talk about the dormant danger, and Stephen gave a great introduction to that concept of low volume disease that's undetectable at the time of surgery. So I'll talk a little bit about how we predict the risk of recurrence in patients with melanoma and what we know about the immune response in these patients. And then I'm going to talk about a novel mouse model that we've been using to better understand the fundamental biology in these patients and how this applies to patients. So I don't need to tell this audience, although on National Skin Cancer Action Week, it's important again to go over that about 14,500 patients were diagnosed with melanoma last year, with almost 2,000 patients dying of the disease. However, most patients are cured with surgery at the time of diagnosis. It might surprise some people to realize how basic the stratifying features are that we use to predict the risk of recurrence for patients diagnosed with new melanoma. In fact, the single most reliable marker is a ruler. So the pathologist takes the ruler and measures how many millimeters the melanoma goes into the skin, and the thicker it is, the deeper it goes into the skin, the higher the risk of recurrence. It's a lot cheaper than ctDNA, but it always surprises me that in 2019, that's the best test we have. But you can see here, this is what I was talking about in my earlier presentation about the AJCC staging system, where we try and stratify patients by risk of relapse. And these lines all represent different risks of relapse for patients with primary melanoma. So this is melanoma that has not spread to lymph nodes, looking at both the thickness of the melanoma and whether or not the pathologist sees what's called ulceration under the microscope. And so you can see there's quite a big difference in outcome from a risk of melanoma mortality of well under 5% for patients with very thin melanomas with no ulceration, to a risk of melanoma mortality reaching 50% for patients with very thick melanomas with ulceration. But we also recommend for many patients who meet a certain threshold of risk, a test called a sentinel lymph node biopsy. And this is a test where we can identify the lymph node that is most likely to contain melanoma and remove it, and then look at it under the microscope. And in about 15% of patients where we look, we'll find small volume of disease within the lymph node, which increases the patient's risk significantly. And we're now using this test to select patients to receive adjuvant therapy or drug treatment after surgery to reduce their risk of relapse. And you can see here that identifying the presence of lymph node involvement will significantly increase the risk of a patient having their melanoma come back again. But another marker that's been investigated, again, this has not necessarily made it into clinical practice, but we do discuss it in our meetings, is the presence of an immune infiltrate within the melanomas, or what we call a TIL infiltrate, the tumor infiltrating lymphocytes. So we can see, this is a melanoma here, and you can see it's very pink because it's not very cellular by comparison to this one where it's very purple, it's full of immune cells. And if you look here, this is patients with primary melanoma, a grade 3 immune infiltrate, so that's a tumor that is chock full of immune cells, has a much better prognosis than one where there are no immune cells inside it, because it tells us that the patient's immune system has recognized some aspect of the melanoma. And this is a what we call a multivariate analysis looking for predictors of lymph node involvement in patients with melanoma, and thicker melanomas and melanomas with ulceration are more likely to contain to have lymph node involvement. But if the patient's melanoma is full of immune cells, it's less likely to have a metastasis, because presumably the immune system has recognized the melanoma and has either eradicated or prevented the melanoma from spreading to the lymph nodes. And we know this is what, and this is a paper by a couple of very clever scientists in the United States who defined cancer using a number of hallmarks. And we know that one of the key hallmarks of cancer, that is, what does it take for a cell to turn into a cancer, is that it has to avoid immune destruction. So if, you know, we think that most of us are making cancers all the time. So as we sit here now, we're all making cancers, but they're all being recognized by our immune system and being eradicated. So it's not actually cancer, because it's only truly a cancer if it's not eradicated by the immune system. And here's a great example of the interaction of the immune system with the tumor. So this is a patient who came to see me a few years ago with a big lump under his armpit, which was a lymph node metastasis. And I went looking for where the melanoma had started from, where the primary disease was, but actually there was no evidence of a primary melanoma. But what I did notice is what's called vitiligo, which is depigmentation of his skin. And interestingly, the depigmentation had started very recently before the lump had emerged. And so this is a well-described phenomenon. And what's likely happened is that the patient has had a melanoma, and the immune system has identified the melanoma and eradicated it. But at the same time, there's overlap between the antigens, or the surface markers in the melanoma, and the antigen in the skin. And so the immune cells that eradicated the melanoma also eradicated the melanin from his skin, which is why he's lost the pigment. But the clone of the melanoma that spread to the lymph node was not eradicated. You can see here his PET scan. This lump here that I can show you with my arrow is the lymph node in his armpit. So the big 5 centimeter lymph node, or 6 centimeter lymph node. And he had surgery. This is a beautiful multiplex immunohistochemistry picture from Eloise Alce from Paul Neeson's lab. And what you can see is the melanoma is full of immune cells. These red and green spots are all immune cells filling the melanoma. But the melanoma also has this blue coating, which is PD-L1, which is the checkpoint that is now used for immunotherapy, which is why the immune cells were unable to eradicate the melanoma. This patient had surgery for his lymph nodes, and it's now 4 or 5 years down the track. I see him every 6 months, and his melanomas never recurred. So again, showing you the positive prognostic impact, even though he had a very large lymph node, seemingly high risk of recurrence, but because of the immune engagement here, he's presumably been successful, his immune system's presumably been successful in eradicating the disease elsewhere. And this is what I was just mentioning, that the primary tumor was eradicated before the lymph node escaped. And we know that melanoma is a very immunogenic cancer, because the average melanoma has a very high, what we call mutation burden, largely because of the fact that most melanomas arise due to UV exposure, and UV is a mutagen that causes a lot of DNA damage. So patients with melanoma often have a lot of mutations, and the more mutations you have in your melanoma, the more likely it is that one of those mutations will cause the melanoma cells to expose themselves to the immune system. And that's why immunotherapy is so effective for many patients with melanoma. And we know that the more a melanoma expresses an immune gene signature, the better the prognosis for the melanoma patient. And we've looked here at Peter Mac in this project that I did together with Paul Neeson, where the work was largely done by Eloise and by Andrew Kolbach, who's a very talented pathologist. We looked at a number of melanoma metastases that were resected from patients and characterized the immune infiltrate. And you can see here 2 different patients. This is one patient where there are no immune cells at all in the metastases. What you can see are a lot of purple dots, which are the melanoma cells, but we see very few yellow and orange dots. By contrast, down here, the melanoma is overwhelmed by immune cells, similar to what we saw in that first patient. And basically, we were able to categorize patients into a number of different groups based on the immune infiltrate, both within the tumor, but also within the stroma, or both within the tumor, the stroma, and the surrounding tissue. So what I'm now particularly interested in, more than what's happening in the melanoma metastasis once the patient actually presents, is what's actually going on before the melanoma metastasis starts to grow. So many patients who have a primary melanoma removed, often without lymph node involvement, will not, if they were going to recur, may not recur for many years. And so the question is, what's going on for all of that time? And then what's the switch that happens that causes the metastasis that's been kept under control by the immune system for so long to then lose control? And to answer this question, I've been collaborating with a very talented scientist, immunologist at the Peter Doherty Institute across the road, Thomas Gebhardt, using this model that he, together with Jason Waithman, have established of an epicutaneous inoculation. So what does that mean? So in most melanoma models that have been used previously, the melanomas are either injected under the skin of the mice or injected into the blood system of the mice to allow it to spread. And that's a little bit of an artificial system and doesn't really give us an opportunity to study melanoma the way that it arises in patients, which is in the skin. In this model, the skin of the mouse is scratched and the melanoma is placed directly onto the skin. So it actually grows in the skin, interacting with the normal cells within the skin in an immunocompetent or a mouse with a normal immune system. And what we see in these mice is that in about 40% of them, the melanoma will start to grow, as you can see here at the top. And this is a melanoma which has artificially been created to express what's called luciferase, that it can be detected using a very sensitive detector. That's why it's colorful. But what we see in about 20% of mice is that the melanoma just sits there as a stable deposit. So it's definitely melanoma. When we remove and look under the microscope, it definitely looks like melanoma, but it's not growing. The mouse's immune system is keeping that melanoma in check. And you can see here, for a number of weeks, these tiny little spots, and this is what it looks like under the skin. There's clearly melanoma cells sitting there under the skin of the mouse. So, and about another 40% of them will never develop melanoma and won't even have the melanoma sitting there stable. It's just completely eradicated by the mouse's immune system. This is by contrast to what I was talking about before, the subcutaneous injections where you inject the melanoma under the skin. All of those mice develop metastases because the melanoma doesn't have the same interaction with the immune cells of the skin. And so one of the questions, what are those immune cells? And this is work that was done by Simone Park, who was a postdoc in Thomas's lab, just as we were starting this, this collaboration to try and identify exactly what's going on here. You can see here's a progressing melanoma, and this is an occult melanoma, which in this particular mouse, for almost a year, this little spot just sat there in the skin completely under immune control. And they were able to show that if you eradicated the immune cells from the mice, then all of the mice would develop metastases, clearly demonstrating that it's actually the immune system that's responsible for keeping these melanoma cells under control. And what are the immune cells specifically? So we think that it's these tissue-resident memory cells that express this marker called CD103, because if you have a mouse where the melanoma progresses, there are very few of those cells, whereas if you have a mouse that is able to control the metastasis, then you have a very high percentage. You can see here 0 versus 87% of these particular cells. And they went to extreme lengths to demonstrate this through some amazing images and videos, which I'll show you. So the red cells here are the melanoma cells, and the green cells here are these CD103 tissue resident memory cells. Hold your camera because I'm going to show you one of my favorite videos. So this is a melanoma that's growing. You can see here a lot of red cells and some green cells, but the green cells are sort of just not really able to properly infiltrate into the melanoma. You can see here there's big areas without any green cells at all. But by contrast, here is a melanoma. Look at this guy here. This is a couple of cells sitting here with these tissue resident memory cells. Really, they've got their arms all over it. It's not letting it grow. It won't let it go. It's like a rugby tackle. And you can see, so these are very successful TRMs keeping that melanoma under immune control. Here's another one. This one, you can see each of the cells has at least 2. This is just in one plane, so if you can imagine if you go up and down, you would have even more of these green cells, but each of the cells has at least 1 or 2 of these tissue resident memory cells, which just won't let it go, keeping it in check. And presumably this is what's going on in our patients all the time. Patients living their lives with a few melanoma cells under immune control for many, many years. So here's one of my patients where we sought to answer the question, does the same cell type exist in patients? So this is a patient who had a melanoma here. You can see a large skin graft on her leg. And here's number 1. So about a year before I met her, she popped up with what's called an in-transit metastasis. So an in-transit metastasis is when melanoma spreads within the lymphatic system in the skin, and patients pop up with a little spot near where the melanoma had started. And the surgeon she saw removed that spot, and again, she was free of disease. And then she came to see me at the time. We talked about whether we should give her any drug treatment. She decided not to have any, and so we just kept a close eye on her. And then she popped up about another 9 months later with a spot here at number 2, very similar to the one that had been there previously. But this time she said to me, let me show you this tiny little red spot over here at number 3, which is how they all start. So the one that she initially came in with was like 3 millimeters, 4 millimeters, brown, clearly melanoma. But the other one was this very, very tiny, like a tiny pimple. And so I said, you know, being the curious surgeon that I am, I said, well, we'll just cut them both out and see what they look like. And so here is the in-transit metastasis. It's a globule of tumour. You can see this purple. This is tumour cells. And then this is the little papule. It's very small, less than 1 millimeter. And when we look at it under the microscope, this is the one that's clearly growing. It's full of brown melanoma cells. These are 2 different melanoma markers that we look at, SOX10 and melan A. And when we look at the immune infiltrate, which are these cells, CD3, 4, 8, CD103, you can see there are definitely immune cells, but they're all being pushed to the periphery of the tumour. There's none of them that have made it into the middle of the tumour. So this tumour has worked out how to evade the immune system. It's growing. It's keeping all those immune cells out of its growing centre. So here's the pink spot. So the pink spot, we'll just zoom in here. You can see just a few melanoma cells. Most of the cells in this are not melanoma cells at all. They're immune cells. And what sort of immune cells are they? So these are the 2 markers. So you can see that the cells express different immune markers. Sorry, these are the melanoma markers. You can see they express different melanoma markers. Some of them are SOX10 positive. Most of them are not melan A positive. But look at this. This is the immune cells. This is CD3. So that whole red spot is full of immune cells trying to eradicate the melanoma. This is, you know, battle royale between the growing melanoma and the immune system. It's really, you know, when you see that that scene from Jaws where the shark pulls the swimmer back under the water and is really trying to get out again. It's pulling back. So here we've got this huge sea of immune cells with a few tiny spots of melanoma inside it. And so what you can see is that most of those immune cells, or a lot of them, are actually these CD103 positive tissue resident cells. And when you look at both CD103 and SOX10, so these melanoma cells, just like we saw in those fancy videos from the mice, most of the melanoma cells are surrounded by these CD103 cells, trying to keep it under control. Now, you might say, well, if there's such good control, why is it growing? And my assumption is, had we not removed this, it would have started to grow. So that's a very good question. I don't know the answer, but this, I think, is the first or the earliest evidence of escape from immune control that we've been able to document in patients with melanoma. So I think it's a really very interesting case. And Han, our Young, is in the audience. He did a lot of this imaging, so thank you, Han. He's very good at using these computers. And what you can do with these is actually you can have the computer analyze the percentage of cells in each of the different lesions. And you can see here that in the melanoma that's growing, we have a lot more CD4 positive cells versus this micrometastasis, or the red spot, which is has a much larger percentage of these CD103 positive cells. The group at the Melanoma Institute of Australia looked at CD103 positive cells in patients with melanoma metastases and showed that the presence of a higher frequency of these tumour cells was associated with an improved prognosis compared to patients with a low percentage of these. So these are clearly important in patients with melanoma that has metastasized. But they also showed that when they gave patients immunotherapy, patients who responded to immunotherapy had an increased number of these cells growing in that first initial biopsy. So these are on-treatment biopsies, and you can see a increased number of CD103 positive cells in the patients who responded. I won't go into these cases, but I'll just turn now to what we're now doing in the lab, which is using this model to try and recapitulate what happens in patients. So when a patient has melanoma, they come and usually are diagnosed by a GP or a dermatologist and get referred to a surgeon to have the melanoma excised. So that's what we're doing in the lab. The mice have the melanoma inoculated onto the skin, and then 2 weeks later, we just excise the area that was inoculated, even if there's no sign of tumour growing there, to see whether the mice melanoma will progress. And we see about 6 different patterns in these mice. The first pattern is that, so this is very, very small. These are mice where the melanoma that's inoculated expressed luciferase. So you can see it on these scanners. This is in vitro scanning. You can see it on the scanner even if it's not detectable clinically, because it's a very small volume tumour that's growing. So you can see the tumour's growing, then it's removed, and then it pops up a couple of weeks later in the lymph nodes. So very similar to what we see in patients. But sometimes what happens is the tumour's inoculated, it's removed, it pops up in a lymph node, but then it goes away. And then it comes back again a few weeks later and starts to grow. So the immune system controls it for a while before it then escapes. What we've also seen is before the primary is removed, it's already spread to a lymph node, but then we cut out the primary and the lymph node disappears. And then the lymph node starts to grow again. Here's one where the lymph node metastasis happened very early at 1 week, was gone by the time the patient or the mouse came for surgery. And then once the tumor was removed, neither the primary nor the metastasis ever popped up. Or this one where the primary was removed and we've never seen any sign of melanoma again. So I think this model of removing the inoculation site really recapitulates all the different patterns of the disease that we see in humans. And what we're now doing is we're using various methods to alter the immune environment in the mice to change the rates of metastasis, or to try and change the rates of metastasis. So you can see here in the IL-15 knockout mice, which don't have effective NK cells, that they have a very high rate of metastasis, or these ones which are perforin knockout mice where the immune cells are not particularly effective, they have very high rates of metastasis by comparison to the wild type or normal mice where we see metastasis in about 1/3 of the mice. And then what we also are able to find in these mice when we eradicate the immune cells completely is that metastases pop up all over the place. Pretty much every organ in the mouse has some melanoma in it once we deplete the immune system, demonstrating the importance of these immune cells in keeping the melanoma under control all over the mouse. And so really there's this immune equilibrium where the battle is ongoing between the tumor cells and the immune cells. Oh yes, sorry, I was just getting so excited I just couldn't stop. And so I'll just conclude there. And these are some things that we're doing into the future. I'll thank all of the excellent collaborators who have done a lot of the work, and my colleagues here in the melanoma service. So thank you very much for your time.
Well, I'm sure I was absolutely fascinated and very excited about your work too. So, so I think we have run over time, but Erin, do we have time for a couple of questions? And we've definitely got someone enthusiastic up the back.
Just because there's a booking afterwards, I really apologise.
So if
anyone does have any questions, maybe if we can exit the theatre and grab Stephen and David.
Okay, great. Thank you so much. And we've also got a small gift for these guys. So we'll